Welcome to Quicksand, the most important news in pharma and biotech this week. Beam Therapeutics, a new Cambridge, Massachusetts-based startup co-founded by gene editing innovator Feng Zhang, has raised nearly $100 million in Series A funding in early commitments, according to reporting from the Boston Business Journal. The journal reported that Beam raised an initial $13 million from Arch Venture Partners and F-Prime Capital Partners. About another $85 million has been committed before any formal announcements, the report said. The news followed last week's unveiling of a new CRISPR company, Mammoth Biosciences, which will utilize gene editing technology from the lab of co-founder Jennifer Doudna, who will also serve as chair of Mammoth's Science Advisory Board. Zhang and Doudna have been embroiled in an international legal dispute over the patents on CRISPR-Cas9 gene editing technology. Last year, the U.S. Patent and Trademark Office sided with the Broad Institute, Zhang's institution, saying the inventions claimed by Broad were distinct from those asserted by Doudna and UC Berkeley. According to the report, Beam is pursuing methods to more precisely edit individual base pairs without snipping the molecule's double helix structure. An additional Zhang effort, Arbor Biotechnologies, recently announced over $15 million in Series A funding and a new CRISPR-associated enzyme, Cas13d, that might allow easier delivery to cells. Mammoth, meanwhile, hopes to develop a CRISPR-based platform to detect DNA and RNA-based biomarkers across a wide variety of disease areas. The company currently lists several investors but has not disclosed the amount of capital it has raised. Details on why Fresenius last month sought to terminate its acquisition of Acorn recently emerged, with Fresenius alleging a top executive at Acorn had knowingly submitted fabricated data to the FDA. Fresenius alleged that an Acorn executive vice president for quality assurance in 2012 knowingly directed the submission of fraudulent testing data to FDA as part of an application to market the antibiotic azithromycin. Fresenius said its investigation revealed blatant fraud at the very top level of Acorn's executive team, stunning evidence of blatant and pervasive data integrity violations. Fresenius last month also publicly announced that its independent investigation found material breaches of FDA data integrity requirements relating to Acorn's operations. Last week, however, Acorn asked the Delaware court to require Fresenius to fulfill its obligations under the $4.3 billion merger agreement, noting Fresenius' attempt to terminate the transaction on the pretext that the findings from the ongoing investigation are a breach of the merger agreement is completely without merit. The previously disclosed ongoing investigation, of which we have voluntarily notified and are in regular communication with the Food and Drug Administration, has not found any facts that would result in a material adverse effect on Acorn's business, and therefore there is no basis to terminate the transaction. The investigation is not a condition to closing, and the only remaining condition is approval from the Federal Trade Commission, Acorn said in a statement. The European Medicines Agency has released a revised guideline on the clinical evaluation of vaccines for consultation. EMA's replacement for text, adopted more than a decade ago, addresses questions that have arisen over that time, including issues linked to the emergence of vaccines that contain antigens from multiple pathogens and interest in prime boost regimens. As EMA notes in its draft guideline, the vaccine landscape and the questions raised by sponsors and its own reviews have changed considerably since the existing document came into force in 2007. The changes have led EMA to completely rewrite the text and expand its scope. The result is a document destined to replace the vaccine guideline and other texts on adjuvants and summary of product characteristics requirements. In highlighting the key changes to the guideline, EMA zeroed in on sections intended to address the development of vaccines that feature antigens from multiple pathogens or multiple subtypes of one pathogen. Typically, EMA wants developers of vaccines that contain novel combinations of antigens to compare the immune responses they trigger to those achieved by separate administrations of the antigens. However, if the vaccine contains a very large number of antigens, this approach may not be feasible. Vaccines that target multiple subtypes of an antigen could encounter this problem. EMA provides no advice specific to this situation, stating only that strategies should be designed case by case. The European Commission launched a public consultation as a part of a study evaluating European Medicines Agency's fee system for approving and monitoring human and animal medicines. Until August 2nd, industry and other stakeholders can offer their input on whether EMA's current fee and remuneration system is cost-based, fair, proportionate, and not unduly complex. Under this current system, EMA is funded by EU and European Economic Area contributions, as well as by fees paid by entities for obtaining and maintaining marketing authorizations and for providing other services. The cost of submitting marketing authorization applications for human medicines typically begins at €286,000, while payments for scientific advice can range from €43,000 to €86,000. This commission consultation aims to elicit information, views, and concerns on the fee system and its implementation, including the remuneration to national competent authorities. In particular, it seeks to gather input from groups having experience with the fee and remuneration system on its effectiveness and efficiency, relevance, coherence, and sustainability, the Commission said, noting that some of the questions are more specifically addressed to the pharmaceutical industry, academia, and research organizations that have active experience with the operation of the EMA fee system. The study, including the public consultation, is being carried out by RAND Europe. Following issues by both regulators and industry in interpreting and implementing the International Council for Harmonization's S9 guideline on the nonclinical evaluation of cancer drugs, ICH has recently adopted a question and answer document to help. The 19-page document includes more than 40 questions and answers, offering more clarity on the scope of the guideline, studies to support nonclinical evaluations such as toxicology studies, nonclinical data to support trial design and marketing, and other considerations. The document also offers clarity situations where one species may be acceptable for toxicology testing. The ICH S9 guideline, nonclinical evaluation for anti-cancer pharmaceuticals, reached step 4 in November 2009, though since then, all the parties using the guideline have experienced some challenges around implementation, ICH said. Implementation of the guideline has revealed areas that are open to broad and divergent interpretation by both regulatory authorities and industry, the Swiss group added. For this reason, ICH formed an implementation working group in October 2014 to develop this Q&A document to provide additional clarity around cancer drug development. The questions and answers developed by the IWG are intended to facilitate the implementation of the S9 guideline and, of additional benefit, to continue progress in the 3 R's of reduction, refinement, and replacement in use of animals, ICH said. Thank you for listening to Quicksand, episode 29, May 7th, 2018.